Ectobacteria-mediated transformation, the first ever DNA delivery method that created a revolution in the history of plant biotechnology. Let's see how the idea has generated. The tumor-forming neoplastic crown gall disease of plants, that means a disease called crown gall that used to occur in the root of some perennial crops, was prevalent in the plant section for over a long period of time. However, until 100 years ago, no one has the idea that through which or by which organism this type of disease was occurring. With the experimental approach of Smith and Townsend, they were successfully found that Agrobacterium tumefaciens is the causative agent for such crown gall disease in perennial crops. A scientist called Armin C. Brown, working at the Rockefeller Institute of New York and a pioneer in crown gall research, had demonstrated the transformed nature of the crown gall tumor cells. That means crown gall is such disease that cause tumor or induce tumor in the plant cell. And after developing tumor, when the newly transformed tumor cells become freed from the bacteria, they show some unique features. And that unique features give them the ability to grow indefinitely for a longer period of time, continuous cell division without the need of nutrient hormones or plant regulatory growth hormones. Whereas for normal and non-transformed cells require media supplemented with plant growth regulators for continued cell division. Then Armin Brown proposed that a tumor-inducing principle present in the bacteria, that means from bacteria the plant cell has acquired such capability or such phenotype of autonomous growth and transformation. The timely availability of the new experimental tools played an important role in the discovery of ivosinin in Schell's group at Ghent, and that the virulent strains of Agrobacterium contained a large extrachromosomal element that harbored the genes responsible for the induction of crown gall. To simply state, there is a plasmid, which we know that its plasmid is an extrachromosomal element in the bacteria, and these plasmid contain a specific gene through which the disease occur. That means this gene has the responsible characteristics to cause tumor or crown gall disease in the plant. Later they found out that it was the megaplasmid or also called TI plasmid, because this plasmid has the ability to induce tumor. That is why they are called tumor-inducing plasmid. The loss of virulence by virulent strains of bacteria grown at high temperature was accompanied by the loss of TI plasmid. They also shows that if they apply heat on the virulent strain of the bacteria at a very higher temperature, there is a loss of the virulence capability as well as the loss of the TI plasmid. So it clarifies that it is the TI plasmid which induces or which is the responsible virulence agent. Only a small part of the TI plasmid, crescent tDNA, that means it is crescent means it is dye. Crescent is a dye. A color tDNA was shown to be responsible for tumor formation. It was present only in the nuclear DNA fraction of tumor cells and was found to integrate at random sites rather than at any specific locus. To summarize, the TI plasmid, which possesses the gene responsible for crown gall disease, at that gene later found on the tumor cells or the DNA of the tumor cells at random location. That means there is no specific site at the host cell or tumor cell where the TI plasmid specific gene portion will bind. That means the gene of the TI plasmid can easily bind anywhere in the plant cell. This is their basic capability, to bind anywhere. So these investigations also made it clear that the capacity of crown gall tumor cells to undergo autonomous growth was related to their capacity acquired as a result of transformation to synthesize plant growth substance and metabolites required for sustained growth. So what is the hidden mystery here? The hidden mystery is that such tumor-inducing plasmid related gene or such portion of foreign gene gave the plant cells the capability to produce their own plant growth substances. That is why they do not require any other external plant growth material. They by themselves create or produce their own metabolite and other plant growth substances required for their growth and can continue or carry out their indefinite cell growth system. This is the hidden mystery why such tumor cells do not require any external nutrient or plant growth hormones. So that was the basic theme of Agrobacterium tumefaciens mediated transformation. Later on, we have known that there were two other methods for DNA delivery. Among them, the second one is direct DNA delivery into protoplast. Remember, it is in the protoplast, not in the intact cell. A lecturer in plant physiology at the University of Nottingham, Edward C. Cocking, was interested in producing isolated cells from plants with the aspiration of developing a plant cell bacterial type of culture system in which plant cells would divide, separate, and produce a culture of single cells. That means he was aspiring to produce a plant cell culture type just like bacteria, where we put a single bacterial colony or loop full of bacteria, and from that we can get or collect some single colonies. Similarly, he was trying to put some plant cell in a certain plant culture petri dish and want to get some single cell or cluster of cells. He successfully isolated protoplast by incubating segments of root and other tissues in a cellulose preparation of Myrothecium verrucaria or infiltrates of fungal cultures containing crude mixture of cell wall hydrolyzing enzyme. So he successfully isolated protoplast by applying just cell wall hydrolyzing enzyme from any source, either from fungal source or other bacterial source. So the isolation of protoplast was successful. Two key developments in 1970 and 1971 pointed to the potential use of protoplast for the plant improvement. At the same time, scientists were working on finding the significance of protoplast in plant improvement. Among them, the two significant discoveries were here. The first one is tobacco mesophyll protoplast. That means from tobacco plant, the mesophyll, which is a leaf cell protoplast cultured by Itaru, a Japanese plant virologist, regenerated cell walls, underwent sustained division and regenerated whole plants. That means from mesophyll protoplast, he were able to regenerate cell wall as well as a whole plant. At the same time, Cocking's group showed that protoplast of diverse species, irrespective of taxonomic relationship, could be fused by chemical treatment. So the first discovery was that from protoplast, whole plant can be regenerated, and again Cocking's group showed that protoplast of different species, irrespective of taxonomic relationship, can be easily fused by chemical treatment. That means we can fuse two protoplasts by applying certain chemicals. Indeed, the principal use of protoplasts has been in the genetic transformation of plants. The absence of the cell wall allows DNA to be easily delivered into protoplast by osmotic or electric shock, which temporarily perturbs the plasma membrane. So it's very simple. When we detach the cell wall, which is the hardest shell of a plant cell, from the cell itself, then it becomes so much permeable for anything to inject. So it is very easy to inject the DNA either by osmotic or electric shock within the protoplast of the plant cell. That was the principle of protoplast induced genetic material delivery. Methods for the direct delivery of DNA into protoplast developed during the early 1980s. Proved to be especially useful for cereals, which are considered at the time to be outside of the host range of Agrobacterium. That means the cereal types of plants where Agrobacterium mediated transformation cannot work properly can be used by direct DNA delivery into protoplast. So it is a plus point for this approach over Agrobacterium mediated transformation. Now the third one, or with the most suitable one, direct DNA delivery into intact cells. Technical difficulties faced with the isolation of long-term maintenance of embryogenic cultures, the only source of totipotent serial protoplast. That means from embryogenic culture, the only totipotent serial protoplast can be gained. However, the maintenance was too cumbersome and long and lengthy, and there were some limitations of Agrobacterium mediated transformation. That is why a third most possible approaches were invented. So the most notable success in these efforts was achieved by John Stanford at Cornell University. He and Adolph, who was an electrical engineer at Cornell, developed the idea of shooting DNA-coated tungsten particles. That means some tungsten particles coated with DNA into epidermal cells of onion with a blast of gun. That means it's just like shooting a particle coated with DNA into the epidermis of a plant cell. through which the DNA can be easily delivered, and the point should be specific. These attempts were initially met with laughter and ridicule. At first, people used to laugh at their such approach of combining electrical engineering approach in biology. However, later on, this became the most successful approach of DNA delivery in plant science. So plant biotechnology has been also advanced considerably by the sequencing of plant genomes such as those of Arabidopsis, rice, and others. During the past few decades, a wealth of new information about useful genes and gene regulation, expression, and function, etc., has a greater impact in case of plant biotechnology. These insights, combined with the availability of molecular maps and markers, has helped plant breeders produce new and useful varieties. Molecular breeding is increasingly becoming an important and powerful tool for plant improvement and variety development.